The last type of study design in analytical epidemiology is randomized controlled trial, or fondly known as RCT. RCT are experimental studies where the effect of an intervention is assessed by collecting data before and after an intervention. RCT is used to compare an intervention with one or more other intervention or with no intervention. Interventions are often clinical treatments but may also be educational intervention, example, health promotion leaflets. There are two main features of RCTs. The first one is they are comparative, and the second one is they are designed to minimize bias. Let's look at the first feature of RCT, which is comparative. In RCT, an intervention is investigated by comparing one group of people who received the intervention with a control group or control arm who do not receive the intervention. Control group receives usual or no treatment, and their outcome measure or the change in measure from the baseline is compared with that of the intervention. This figure shows the RCT design. When the study begins, we call it as the baseline point here, and the study population is randomized at baseline, randomized into either intervention group or control group. Randomization means that the person or the researcher has no control in assigning an individual into an intervention or control group, and the assignment is done randomly. And at the end of the intervention, the outcome is measured and reported as there is outcome or no outcome among intervention and control group. The second feature of RCTs is minimizing bias. There are 5 types of bias in research that can be minimized by using RCT. The first one is allocation bias. Second one is performance bias. Third one is assessment bias. Fourth one is attrition bias. And the fifth one is allocation concealment. Allocation bias occurs when the measured treatment effect differs from the true treatment effect because of how participants were selected into the intervention or control group. In RCT, participants will be randomized to either an intervention or control group at study entry. Randomization ensures that characteristics that might affect the relationship between the intervention and outcome measures will be roughly equal across all arms of the study. Therefore, this minimizes potential bias. On the other hand, performance bias occurs when participants' response to the treatment is affected by knowledge of the group to which intervention they are assigned. That is, they know which group they belong to, either intervention or control. Performance bias might also occur when health professionals administer treatment differently between treatment arms. The third type of bias in RCT is assessment bias. Health professionals assessing the outcome of treatment relative to alternative or placebo interventions may record outcome measured biased by their knowledge of the group assignment. Overestimation or underestimation of the effects of an intervention is known as assessment bias. There might be a systematic difference in measuring the outcomes between two groups because of the method of recording used. Example, control group is assigned to one practitioner and the intervention group to another practitioner, or groups are assessed at different times of the day. How to minimize the assessment bias? First one is use a standardized method or evaluation across both groups of intervention and control. The second method is to avoid using subjective measures to assess the effectiveness of a treatment which are more prone to bias. The fourth type of bias in RCT is attrition bias. Attrition bias is also called as loss to follow-up bias. Attrition bias occurs when patients drop out of the study from their respective study group. If halfway through a study the treatment has been successful, participants may drop out and information about the success of the treatment is then lost. Participants in the control group might also be unhappy with their lack of progress and may drop out of the study in order to seek alternative help. When we do allocation concealment, bias will be minimized where the allocation schedule is concealed of whom is assigned to which group, either intervention or control. Blinding or masking helps prevent systematic differences between comparison groups in prognosis or responsiveness to treatment. Blinding of both participants and practitioners prevents performance and assessment bias by ensuring everybody, including participants, treatment administrators, those measuring outcomes, do not know which treatment was given. It is recommended RCT participants are blind to the treatment they receive because control group received a placebo, which is a similar treatment to the intervention. Now we know RCT is challenging, but why we need to carry out RCT? RCT are prospective longitudinal studies which allows causal association between intervention and the outcomes. The random selection of participants into each arm and the controlled way in which trial is carried out mean that all factors are considered equal. Another important point is other study design cannot infer causality like what RCT can do. Now, let's look at 5 important factors in RCT. These 5 important factors are sample size, stratification, trial design, between group contamination, and finally, ethical issues. Sample size depend upon the power of the test, what size of intervention impact is considered meaningful, and the type of hypothesis the RCT is testing. The smaller magnitude of difference between groups that is to be detected and the greater the variability in the outcomes, the larger the sample size that will be required. In terms of stratification, very large trials are likely to have a good balance of patients within each arm. When samples are small, however, treatment groups may be chance end up with different characteristics, and this may affect the outcome trial. Stratification is a way of ensuring the treatment groups are balanced on characteristics that are likely to alter the relationship between treatment and the outcome. There are two commonly used trial designs to allocate treatment and control regimens in RCT. The first trial design is parallel design and second trial design is crossover design. In parallel design, different patients will be randomized in each treatment group. There will be differences in characteristics of the participants. This figure shows the parallel design where the population is randomized into placebo, treatment arm number 1, and treatment arm number 2. You can also have only two groups, which is placebo and treatment arm. Crossover trials are another way of overcoming differences in groups by keeping the patients as matched as possible. Instead of having different patients in each treatment group, patients receive first treatment and then the other treatment in random order with a washout period in between. So, the patients will be randomized into placebo or treatment group for period number 1. For those who receive placebo, they will then undergo a washout period before going to active treatment group. So the same patient will be their control in this crossover design. Within patient differences are then compared in crossover design. Each patient effectively becomes their own test and control. The fourth important factor in RCT is between group contamination. Educational interventions are prone to contamination. Example, a member of the control arm is a friend of a patient receiving the low-fat diet advice intervention. Therefore, information will be passed between the two arms of trial and thus alter the results. The suggestion is to use cluster sampling because a natural cluster such as geographic areas are randomized in cluster sampling rather than individuals. Therefore, each cluster will be the intervention or control. The final important factors in RCT is ethical issues. RCT is not always possible because of ethical issues when assigning patients to study arms. If one group of patients receive treatment thought to be effective, while another group does not, the ethics of a trial may be brought into question. Similarly, there are some trials that cannot be carried out because they may actively encourage unhealthy practices, example smoking. People cannot be randomized into smoking and non-smoking group. Let's now see the analysis of data of RCT. RCTs are experiments set up to test hypotheses. The null hypotheses in RCTs are usually the intervention will have no impact on the outcome measure. In other words, outcome will be similar in both the test and control groups. The alternative hypotheses in RCTs are intervention will have meaningful effect and statistically significant between intervention and control group. The following are the common statistical method used in RCT. The first one is pre and post intervention differences, which we can use paired t-test. The second method is mean differences pre and post between group. This can be done using independent t-tests. And finally, mean differences pre and post between more than two groups can be done using ANOVA. There are also other statistical methods which is advanced in analyzing RCT data. The advantages of RCT is in its ability to make causal inferences mean that RCT provide the strongest empirical evidence of a treatment's efficacy. Randomization of participants to the test and control arms and concealment of their allocation ensures that allocation bias and confounding or unknown variables are minimized. The study can also be tailored to answer a specific question. However, there are some disadvantages of RCT. High dropout when the intervention has undesirable side effects or there is little incentive to stay in the control arm. Ethical consideration may mean that a research question cannot be investigated using RCT design. For a descriptive overview, it may be cheaper and easier to use an observational design. And finally, prior knowledge is required for sample size calculation for RCT, especially on the level of improvement that is clinically meaningful and the expected variation of improvement in the sample. With that, we have covered all study designs in analytical epidemiology and descriptive epidemiology. Thank you.